All right, we're going to talk about section 3 of chapter 4 on biology of the cell and McKinley text about membrane transport. First of all, you should be able to, after this section, summarize the general process of diffusion, distinguish between cellular processes of simple diffusion and facilitated diffusion, define osmosis, define osmotic pressure, describe the relationship between osmosis and tonicity, compare and contrast primary and secondary active transport, explain the difference between exocytosis and endocytosis, describe the endocytotic processes of phagocytosis, pinocytosis, and receptor-mediated endocytosis. Starting with first with the plasma membrane, which we've spoken about before, serves as that physical barrier between the cell and the fluid that surrounds it, the interstitial fluid and all the extracellular fluid. It regulates movement into and out of the cell. It establishes and maintains an electrochemical gradient, and it functions in cell communication. As far as membrane transport, which is really the process of obtaining and eliminating substances across the plasma membrane, there are two categories. We have passive and we have active. So for membrane transport, for the passive processes, which don't require any expenditure, no cellular energy, no ATP, substances move down the concentration gradient from higher concentration toward lower concentration. Diffusion is the movement of solutes, and osmosis is the movement of water across that semi-permeable membrane. And as far as diffusion goes, there are two categories of simple and facilitated diffusion. Simple, there's no transport protein required for the transport. And in facilitated diffusion, transport proteins are required. And then from a standpoint of facilitated diffusion, there are two categories, channel-mediated and carrier-mediated. The channel-mediated is that ions move through the channel, and the carrier-mediated is small polar molecules are moved by the carrier proteins. For membrane transport that's active, requiring expenditure of cellular energy, or ATP, it involves either the movement of substance up its concentration gradient or the formation or loss of a vesicle when it wants to go the way, not necessarily from high to low, but coming low to high. That said, down the gradient refers to going from high to low concentration. For active transport, we have the ion or small molecule moved against its concentration gradient, and vesicular transport involves a vesicle. For the two categories of active transport, we have primary active and secondary active. Primary active is the energy source from ATP. Secondary active is the energy source from movement of another substance. And as far as secondary active goes, when using another substance for assisting energy and movement, we have symport and antiport. Symport is two substances are moved in the same direction, whereas antiport, we have two substances that are moved in opposite directions. And from vesicular transport under active processes, we have exocytosis and endocytosis. And exocytosis is the vesicle is releasing its contents from a cell, and endocytosis, vesicle is formed as material is brought into a cell, right? Exo, it's exiting, endo, coming in. And the endocytosis, there are three different forms. We have phagocytosis, which is cellular eating, pinocytosis, which is cellular drinking, and receptor-mediated endocytosis, where a receptor is required. So a little more on each one of those. For the passive processes of membrane transport, again, doesn't require energy, depends on substances moving down the concentration gradient from high to low concentration, and the two types are diffusion and osmosis. Active processes require energy, substance must be moved up its concentration gradient, meaning to go low to high, and membrane vesicle must be released in vesicular transport. Diffusion, which is the net movement of a substance from an area of greater concentration to an area of lesser concentration, is of course passive, and molecules and ions in constant motion due to kinetic energy. If it's unopposed, diffusion continues until substance reaches equilibrium. It's going to keep going until there's a balance, and molecules are evenly distributed throughout a given area. The rate of diffusion is going to depend upon how steep that concentration gradient is. If there's this big discrepancy between high and low concentration, it's going to be faster. Okay? And the temperature, of course, plays a role as well. That reflects the kinetic energy or the random movement. The higher the movement, higher because we're due to higher temperature, and a faster rate of diffusion as well. So if you take basically a dye and you put it into water here in a beaker, it's going to diffuse, and it diffuses until it reaches equilibrium. As far as passive processes of diffusion, I should say null diffusion is active, to be clear. We're talking about active diffusion before. Simple diffusion, basically, I'm sorry, it's always passive, excuse me. Molecules pass between phospholipid molecules, try not to confuse you too much. Small and nonpolar solutes, which includes the respiratory gases, oxygen and carbon dioxide, some fatty acids, ethanol, urea. It's not regulated by the plasma membrane. Movement is dependent upon the concentration gradient, and it continues to move as long as gradient exists. And then here we have just a little bit of graphic representation of the simple diffusion of solutes, moving down the concentration gradient from outside the cell to inside the cell, from the interstitial fluid through the phospholipid bilayer, the membrane, into the cytosol of the cell. And over here is showing gases be going up the concentration gradient on the other side over here. Facilitated diffusion, transport processes for small charge or polar solutes, requires assistance from the plasma membrane proteins. We have two types, the channel-mediated diffusion and the carrier-mediated diffusion. In channel-mediated diffusion, movement of the small ions through water-filled protein channels. Channels are specific for one ion type. Only one ion is going to be able to move through that channel-mediated diffusion channel, okay, through that channel for mediated diffusion. Leak channels are always open, and gated channel is usually closed, but it opens in response to a stimulus for a fraction of a second, okay? And that's also, these channels are very important. These channels that are mediated for diffusion are important in normal function of muscle and nerve cells. For carrier-mediated diffusion, small polar molecules are assisted across the membrane by a carrier protein, okay? There's a binding of a substance which causes the change in the carrier protein to change shape. Substances are released on the other side of the membrane, and substances move down their gradient. Uniporter is a carrier transporting only one substance. The number of channels and carriers determines the max rate of substance transport. Okay. So here we can see carrier-mediated diffusion. We have here glucose traveling down that concentration gradient with the assistance of a glucose carrier protein, okay? Because glucose is usually a little bit more larger molecule, needs a little assistance to come through that plasma membrane, unlike sodium and some other substances that are not as large, relatively speaking, to travel through that membrane. Okay. Osmosis is movement of water, not solutes. It's passive movement of water through selectively permeable membrane. The membrane allows passage of water, but the membrane prevents passage of most solutes. That's important to understand due to the polar and non-polar tails and the polar heads, right? The differences in water concentration on either side of a membrane will play a role in how much water change takes place, or movement. Now, there's two ways that water crosses that membrane. It slips between the molecules of the phospholipid bilayer, which certainly happens to some extent, or it moves through the integral protein water channels like aquaporins. Two types of solutes. We have the permeable solutes. They pass through the bilayer because they're permeable, right? Small non-polar solutes like oxygen, carbon dioxide, urea, and non-permeable are prevented from passing through the bilayer. They're charged, or they're polar, or they're larger, essentially, like glucose and proteins. So it's not just whether they're larger, to be clear. Polar or charged will play a role. They're charged specifically, right? The polar heads, if they are non-polar, they're going to play a role with trying to get through, versus non-polar small solutes, they're going to have no problem getting through that layer, but the polar heads are going to have an issue with the polar if they're larger and or charged. The concentration gradients across the plasma membrane between the cytosol and the interstitial fluid, solutes are prevented from moving across that bilayer, and that, of course, water concentrations then are going to be basically causing water concentration differences to exist, right? And the greater concentration of solutes contains lower concentration of water, right? More concentrations of solutes, lower water concentration, higher water concentration, lower concentration of solutes. As far as the movement of water by osmosis, depends on that concentration gradient between the cytosol and the solution. And again, the water is going to move down the gradient until the equilibrium is reached. It moves from the solution of 1% solutes into a solution containing about 3% solutes, okay? It's going to be higher water to lower water, right? As opposed to lower concentration to higher concentration of the solutes. Let's get a closer look in here. Look at osmosis in cells. And so we see the aquaporin here, and we see basically the movement from the inner part of the cell, the cytosol, into the interstitial fluid. Permeable to water, that layer is that phospholipid bilayer. Non-permeable to most solutes if they're charged or polar or large, right? Not going to travel through. That concentration gradient is going to move from a standpoint of osmosis, higher water concentration to lower water concentration. So it's going to be lower solute though and higher solute. Make sure you understand that difference. For osmotic pressure, it's the pressure exerted by the movement of water across the semi-permeable membrane. It's due to a difference in the solute concentration, right? The steeper the gradient, more water moves across. Hydrostatic pressure is the pressure exerted by a fluid on the inside wall of its container. Clearly, the greater level of water or water pressure, I don't think it's going to be affect that gradient, affect that movement as well. And for the osmotic pressure here, see the higher solute concentration, with a lower water concentration, the semi-permeable membrane right here with a lower solute concentration, higher water concentration, right? And it's going to move again from that higher water to lower water concentration. For osmosis and tonicity, a cell gains or loses water with osmosis along with a change in cell volume and osmotic pressure. Tonicity is the ability of the solution to change the volume or the pressure of a cell by osmosis. And the relative concentration of solutions are isotonic, hypotonic, and hypertonic. Isotonic solution is where both the cytosol and the solution have the same relative concentration of solutes. That iso, basically, lack of movement there. It's not high, not low, it's between. An example is normal saline with a concentration of 0.9% sodium chloride, which is commonly used in IV solutions. And there's no net movement of water, okay? Whereas a hypotonic solution has hypo being low, has a lower concentration of solutes, higher concentration of water than in the cytosol, like erythrocytes or red blood cells, and pure water. Water moves down the concentration gradient from outside cell to inside, okay? Going from basically, when we're talking about water, is going to be moving in terms of being hypotonic, right? Increase the volume and pressure of cell. Lysis has to do with, or lysis, if you like, hydrolysis, right? They'll concept this term lysis, or that part of the word for hydrolysis, is the rupturing of red blood cells occurs if difference is large enough, okay? Because they're always breaking apart. Lysis refers to breaking apart. Hemolysis is rupturing of erythrocytes. Hypertonic solution, solutions with a higher concentration of solutes than the cytosol, right? Because we're talking about hypertonic solution here. The erythrocytes in a 3% sodium chloride water solution would be an example of that. Water moves down the concentration gradient and moves from inside the cell to outside, and decreases the volume and pressure of the cell, and with crenation, the cell shrinks. That's what crenation is. So we have an isotonic solution with interstitial fluid is the same concentration as the cytosol, so there's no net movement with normal erythrocytes. And when erythrocytes are nearing hemolysis, we have a hypotonic solution here where interstitial fluid is less concentrated than the cytosol, so it's moving inward. And a hypertonic solution, water is going to leave the cell. Interstitial fluid is more concentrated than the cytosol, so water is going to be leaving to go into the area of higher concentration of solutes but lower water concentration, and that's when erythrocytes are undergoing crenation. Active processes are organized into active transport and vesicular transport, and for the active transport, it's movement of a solute against its concentration gradient, right? Like ATP would be an example, maintains that gradient between the cell and interstitial fluid, and the source of energy determines whether the movement is primary or secondary. For a step of primary, now we're talking about ATP, the breakdown of ATP. Phosphorylation, the phosphate group is added to the transport protein. This changes the protein shape and movement across the membrane. Whereas ion pumps, cellular protein pumps that move ions across the membrane. We have, it maintains the internal concentration of the ions. Calcium 2+ pumps in plasma membrane of the erythrocytes. Through in here from the end of the interstitial fluid, or from the interstitial fluid outside, say it moving outwardly here in this case. This prevents cell rigidity from accumulated calcium. That's important. And the erythrocytes remain flexible enough to move. Sodium potassium pump is going to come up a lot for us, especially when we start talking about the muscles. It's the exchange ion pump. I want to make sure you have a good understanding here. It moves one type of ion into the cell against the gradient, while moving another type of ion out of the cell against the gradient. The plasma membrane preserves the steep gradient differences, essentially. And it continuously exports sodium out of the cell and moves potassium into the cell. That's pretty common and happening all the time. Sodium potassium pump. So here we have 3 sodium ions and the ATP that are binding to the sites on the cytoplasmic surface. Let me get a little closer look here for you here. Starting here like with step 1 of this process here, we have essentially 3 sodium ions, okay, and the ATP bind to the sites on the cytoplasmic surface of the sodium potassium pump. And then the ATP is then split into ADP and the phosphate, okay, and that results in both the binding of the free phosphate to the pump itself, and that release of energy and causes the sodium potassium pump to change conformation, change its shape, and release the sodium ions into the interstitial fluid outside the cell. And then the two potassium ions from the interstitial fluid, okay, outside the cell now here, then bind sites on the outer cellular surface of that sodium-potassium pump. At the same time, that inorganic phosphate that had bound to that pump is now released into the cytosol. The transport protein then reverts back to its original shape, resulting in the release of the potassium ions into the cytosol, and the sodium-potassium pump is now ready to begin the process again and start anew. And of course, then you have binding of ATP inside the cell. Okay. That simple little concept is very important to understand ongoing. As far as active processes for secondary active transport, we're talking about moving a substance against the concentration gradient, using energy for movement of second substance down its gradient, and it provides power to pump other substances. Sodium moves down that concentration gradient, depending on the sodium-potassium pumps for energy. The two types are symport and antiport. With symport, substances moved in the same direction, right? It's also called symport secondary active transport. When they move in opposite directions, they're antiporters, and it's called antiport secondary active transport. Okay, and here's a good example of that, where you can see here with the symporter, substances are moving in the same direction, with glucose transported up its gradient into the cell itself, okay, and from the interstitial fluid to the cytosol and across that plasma membrane. And then we have the antiporter, where the substances are moving in the opposite directions. So we have basically sodium coming down here, and we have hydrogen transported up its gradient out of the cell. Vesicular transport, also called bulk transport, involves energy input to transport those large substances across the plasma membrane via vesicle, and these membrane-bound sacs are filled with materials, and that breaks us down into exocytosis and endocytosis. Exocytosis, the large substances are secreted from the cell. Macromolecules are too large to move across that membrane. They're packed with intracellular transport vehicles. It requires ATP, and the contents are released to the outside of the cell following fusion, like the example of release of neurotransmitters from the nerve cells. So we can see here the vesicle nears that plasma membrane, okay? The vesicle membrane then fuses with that plasma membrane. That's going to open up to the outside of the cells. That plasma membrane opens itself up to the outside of the cell. And that releases the vesicle contents into the interstitial fluid and integration of vesicle membrane components into the plasma membrane. Endocytosis is the cellular uptake of large substances from external environment. We're talking about similar to exocytosis, but it's in the reverse, right? It's for uptake of materials for digestion, retrieval of membrane regions from the exocytosis, and regulation of membrane proteins composition to alter cellular processes. Okay. And three types are phagocytosis or phagocytosis, pinocytosis, receptor-mediated endocytosis. We're talking about cellular eating for phagocytosis, when a cell engulfs a large particle, okay, forms large extensions called pseudopodia, and they surround the particle and then close in that sac and then digest after fusing with the lysosome. When a white blood cell, a phagocyte, essentially engulfs and digests a microbe. There you can see those extensions coming out and engulfing and digesting that substance, okay. Cellular eating. Pinocytosis, cellular drinking. It's an internalization of droplets of interstitial fluid containing dissolved solutes. Multiple small vesicles are formed in most cells. You can see here where multiple small vesicles are formed here for cellular drinking in this case. Cells got to feed and cells got to drink, right? And the receptor-mediated endocytosis uses receptors on the plasma membrane to bind molecules with interstitial fluid and bring the molecules into that cell. It's going to enable the cell to obtain bulk quantities of substances, the transfer of cholesterol from blood to a cell, and of course that cholesterol is bound to a low-density lipoprotein, LDL, right? They move from the blood into the interstitial fluid, then they bind to LDL receptors in the plasma membrane, and then they're internalized. And then you can see, in this case, that ligand receptor complex, which we'll come back to, of course, later on too, a receptor there with interstitial fluid here, and when they're talking about receptor-mediated endocytosis, we'll see that plasma membrane open up here, creating this clathrin-coated pit, okay, and it'll coat that vesicle. This is more for your own reference, just for future reference. For down the road, we'll talk more about these things. You should be able to check yourself for these questions here to see if you're following the content well. Just pause this and going to go through these. And that's chapter 4.3. We'll come back for 4.4 in a moment.